Hello everyone, today we'll be talking about two genetic disorders that resulted from mutation, sickle cell anemia and Tay-Sachs disease. We have Yankai, Emilia, Elias, Rahana and Vaibhav working in this team. Our objectives will be the overview of sickle cell anemia and Tay-Sachs disease, their global and local statistics, the group of people that are at risk, the pedigree chart for each disease, possible effects to the mutation, probability of the family members at risk, and last but not least, the conclusion.
The genetic disorder, sickle cell anemia, causes the normal red blood cells, which are disc-shaped, to appear as a sickle, which could promote blocking in the blood vessels and prevent proper transportation of blood throughout the body. This will lead to probable tissue damage, fatigue, and pain in the arms and legs of the person with the disorder. Sickle cell anemia is an autosomal recessive genetic disorder that is passed down through offsprings. This would mean that a set of 2 copies of the gene are required in an individual for it to be expressed and to have the sickle blood cells. Sickle cell anemia is the result of the substitution point mutation on chromosome 11, which is responsible for the beta hemoglobin protein chain. Looking at the statistics, in America, 300,000 infants are born with the disease annually. 1 in 500 Americans are affected, and 1 in 12 African Americans are at least carriers of the disease. In Malaysia, however, 1% of the Malaysian population, or roughly 300,000 plus, of the population have the disease as carriers. When talking about the susceptibility, there are no age groups where the sickle cell anemia will appear and disappear, since it's a genetic disorder and it's not as simple as a common flu. The prevalence shown amongst age groups of young children up to elderlies who range more than 50 years old or at 50 years old show no real significant difference in the prevalence, which would roughly range from 6.4% to 7.4%. The only key different aspect to acknowledge is that elderlies may be more susceptible to the symptoms of the disorder due to their weakened immune systems and bodily functions. People from regions that have endemic malaria are more likely to be carriers. This includes people from Africa, India, Saudi Arabia, as well as the Mediterranean. And from here, the pedigree tree shows how the genetic disorder will be passed on from parents to offsprings, where females would need 2 copies of the genes for it to be expressed, and males will only need 1 copy of the gene for it to be expressed.
According to the Dina and her family's pedigree, Dina is the carrier of the disorder. This indicates that her offspring will have a 50-50 chance of being normal or carriers of the disease if her partner is free from sickle cell anemia disorder. Meanwhile, if she marries to a disease carrier husband, their kids will be 25% normal, 50% carriers of the disease, and 25% inherited the disease. Sickle cell anemia is caused by germline cell mutations. It is possible that it will be passed down to the offspring, and if the generation continues to reproduce, it will be a never-ending cycle. Sickle cell anemia is an autosomal recessive illness, which means it may be passed down to kids but may skip a generation, since it requires 2 defective alleles, 1 from each parent. Otherwise, the offspring will simply be a carrier of the disease or a normal individual. Besides that, sickle cell anemia is caused by a single mutation in the beta-globin gene. On both homologous chromosome 11, there is a single point mutation in exon 1. The 17 nucleotide thymine was substituted for adenine, inverting the TA pairs to an AT pair, resulting in an irregular hemoglobin with valine instead of glutamic acid at the 6th codon. When the negatively charged glutamic acid is eliminated, electrophoretic mobility is also affected, and when the oxygenated valine enhances hydrophobic contact with other sickle hemoglobin molecules. As a result, sickle hemoglobin molecules cluster in large numbers in the erythrocyte, generating hemoglobin polymers that affect the architecture of the red blood cell, a condition known as sickling. The amount and time of hemoglobin deoxygenation, as well as the intracellular sickle hemoglobin concentration, are proportional to the degree and extent of sickle hemoglobin polymerization. Sickle erythrocytes, on the other hand, are rigid and prone to lysis, as well as interacting with leukocytes at the vascular endothelium. As a result, hemolytic anemia occurs, and so does recurrent small vessel blockage. Vessel occlusion induces tissue ischemia, which causes significant pain and organ damage over time. Chronic inflammation is caused by increased reactive oxygen species formation following reperfusion of ischemic tissues. Inflammation also causes an increase in the synthesis of adhesion molecules, leading sickled erythrocytes to attach even more strongly to the artery wall and worsening vessel occlusion. To treat the sickle cell anemia condition, numerous repair strategies are available. Blood and exchange transfusions are one of the most popular mechanisms. Transfusions of healthy and normal red blood cells stimulate a rise in hemoglobin level in sickle cell anemia patients. As a result, it has the potential to lower the amount of abnormal red blood cells in patients. Furthermore, blood transfusions lower the percentage of sickle hemoglobin erythrocytes, limit their formation, and hence minimizes hemolysis. It also reduces the chances of vessel occlusion and ischemia. Blood transfusion can be simple top-ups or exchange transfusions, in which blood is withdrawn and restored at the same time. Exchange transfusions successfully decrease sickle hemoglobin erythrocytes and the danger of iron excess. This repair mechanism, however, is required for larger volume of donor red blood cells to function.
Tay-Sachs disease is an autosomal recessive disorder caused by a mutation in the gene hexa which encodes the enzyme beta-hexosaminidase A required to catalyze the breakdown of a fatty substance known as GM2 ganglioside. The symptoms of the Tay-Sachs disease are sight and hearing problem, seizures, slow mental and physical development, gradual loss of mobility, muscular atrophy leading to paralysis, exaggerated startle response, and dementia. Tay-Sachs disease is rare in the general population and the incidence is about 1 in 100,000 live births, whereas the carrier frequency is about 1 in 250. The groups that are mostly at risk are people who are descendants of Ashkenazi Jewish, French Canadian, and Louisiana Cajuns. Tay-Sachs disease manifests in a wide spectrum. Infantile Tay-Sachs is a prototype of degenerative disease of gray matter in infancy. The infant is usually normal at birth and the symptoms start between 3 to 6 months but can manifest as early as 1 week of life. Juvenile Tay-Sachs disease manifests in early childhood between ages 2 to 10 caused by the reduced activity level of HEX-A. Adult Tay-Sachs disease is an infrequent disorder and diagnosis is often delayed at least by 8 years of age. The adult form is less aggressive, resulting from small mutation and high residual activity of HEX-A. Typical symptoms start at adolescence or early adulthood but can appear later.
The diagram on the left shows the timeline of how somatic mutations come to be and present themselves within an organism. This slide shows the pedigree chart of an individual with family history of Tay-Sachs disease. The proband, nicknamed Alex, is neither affected nor a carrier of the gene mutation. Germ cell or germ line mutations occur within either the male or female gamete. This means all cells within the affected individual contains the mutation. Germline mutations can be confirmed, as these can be inherited by offspring. Somatic mutations only occur after the zygote is formed, and only affect one cell originally. Since only non-reproductive or somatic cells can be affected, the mutations and disorders or diseases derived cannot be inherited. Based on the pedigree chart seen previously, we can conclude that Tay-Sachs disease is caused by a germ cell mutation rather than a somatic mutation.
Tay-Sachs disease is caused by a mutation in a gene hexa located at 15q23. More than 130 mutations have been identified so far and it includes single gene deletions, substitution, insertion, splicing alteration, duplication, and complex gene rearrangements. Absence of vital HEX-A enzyme results in abnormal accumulation of GM2 ganglioside in the nerve cells of the brain and the spinal cord. This ongoing accumulation causes progressive damage to the cells. This is an example of a mutation that occurred in the hexa gene. The mutation shown is a frameshift mutation where 4 additional nucleotides were inserted, resulting in Tay-Sachs. There is no cure or effective treatment for Tay-Sachs disease, however, researchers are pursuing several approaches to finding a cure. Scientists are exploring enzyme replacement therapy to provide the HEX-A that is lacking in babies with Tay-Sachs. Enzyme replacement therapy is a promising option for Tay-Sachs disease. Nevertheless, currently ERT is less effective in Tay-Sachs disease due to the inability to cross the blood-brain barrier and preventing neurological complications. Enzyme-enhancing therapy provides strategies to reduce the substrate with the use of molecules called chaperones to stabilize the enzymes. The rationale behind SRT is balancing substrate synthesis with that of diminished enzyme degradative power. Since Tay-Sachs is caused by a single gene disorder, gene therapy will be an excellent option. Great progress has been achieved so far by developing adeno-associated virus-based factors. The transplantation of ex vivo modified multipotent neural cells with human hexa gene expression produced by retroviral transduction is another major breakthrough approach.
Hemorrhage and affected women who can pass the disease to the next generation. If Alex's brothers decide to have children instead, 50% of them would be a carrier of the disease, while 25% would be affected, and the other 25% would be normal, would not carry disease or have it. On the other hand, if Alex's sister decides to have children, and she marries a heterozygous male, half of the offspring would be carriers, while the other half would be affected. However, since Alex's sister is homozygous for the disease and decides to marry someone who is homozygous as well, then all the children will be affected. Tay-Sachs disease, or TSD, can be classified as an autosomal recessive disorder and is caused by a mutation on the hexa gene. TSD can be caused by the absence of a vital enzyme called hexose imidase A, and therefore fatty substances cannot be broken down that as a result accumulate in the nerve cells. Both genders are affected by this, and it is not sex-linked disorder. However, symptoms and severity of the disease vary with age. People with TSD usually suffer from paralysis as nerve cells and spinal cord are affected. As for sickle cell anemia, this is caused by a mutation that occurs on chromosome 11. It is a type of point mutation where there is a base substitution from glutamic acid to valine. As for sickle cell anemia, normal red blood cells are distorted to a sickle-like shape due to a type of point mutation. In this point mutation, glutamic acid is converted to valine. This congests circulatory systems like veins or arteries, and this results in organ damage. Symptoms usually vary in severity, and this disease can be detected by carrying out hemoglobin electrophoresis.